Hi everybody, welcome to Lab 7. Today we'll be looking at caffeine determination in foods and beverages using HPLC DAD. That stands for High Performance Liquid Chromatography with a Diode Array Detector. First we're just going to do a little overview of caffeine, then we'll get into the reasons we use HPLC and what's going on when we use an HPLC. Unfortunately, due to university shutdowns, the actual lab footage is a little bit limited in this lab. So we'll cover what we can in this pre-lab section. I do have a little bit of footage of an injection and an elution of the compounds. And then we'll go over what a report looks like coming out of the HPLC and how to interpret that data. And then you'll go ahead and do your reports based on the raw data we provide. So caffeine is a part of a group of compounds known as methylxanthines, and so here are the three methylxanthines that you might have heard of. We have caffeine, which we know is a stimulant. Theobromine is predominantly found in chocolate, and then we have theophylline. So theophylline is actually a bronchodilator, and it's used to treat conditions like asthma and COPD. So they're all very closely related compounds, and all have physiological effects. So caffeine is a GRAS product that's generally recognized as safe. If it's added, you must list it as an ingredient in your product, but you don't actually have to do quantitative labeling. So it's actually voluntary. And the reason we see that so often is because there's guidelines by the American Beverage Association suggesting how to label caffeine. So normally you see milligrams per serving on a label. One thing we do need to be concerned about with caffeine is that it is a potential toxin. So at low levels, it gives us this alertness, but at relatively high levels, it can be toxic to humans. It can be acutely toxic, so it can cause death, but at lower levels, you will get effects like headache, nausea, sleeplessness, overexcitement, and palpitations of the heart. And so we're seeing that at levels that are 0.5 to 1 gram, so 500 to 1,000 milligrams. And so that would be like several cups of coffee. If one cup of coffee is usually around 85 milligrams, you have to consume quite a bit of a natural product to get that effect. But when we talk about more concentrated sources of caffeine, like energy drinks or caffeine pills, you might start to experience some of these toxic side effects. And then when we get up even higher, up to 10 grams or more, we can have serious issues like delirium, hypertension, arrhythmia of the heart, and eventually death. So quantifying caffeine, why do we want to quantify caffeine? Like I said, it's a potential toxin and there's a lot of sources. And if we're making a beverage, we want to be able to label our beverage with the amount of caffeine in it so that our consumers can track how much caffeine they're taking in from all these different sources. And so we'll ask, what kind of approaches can we take? We've quantified a lot of things so far in this semester, and maybe you'd say, what if we just quantified its absorbance versus a standard concentration of caffeine? And if we look at this absorbance spectrum of caffeine, it looks like that might work. So we have a maximum absorbance of around 275 nanometers. So maybe if we just looked at the absorbance of a beverage at 275 nanometers and quantified that against a standard curve of caffeine, we could get a quantity of caffeine. And maybe that'd be fine if we're just looking at something like caffeine and water. But the reason it's so challenging to quantify caffeine is, let's say we take a beverage like coffee. So if coffee has a bunch of different compounds in it, it's just a biological mess. And we could have a bunch of different compounds, each with these different absorbance spectras that overlap with caffeine. So now if we put this in a cuvette and try to measure the absorbance at 275, we have a lot of different signals kind of coming in and convoluting our results and artificially inflating the amount of caffeine that we'd be measuring here. So what we need to do is find a way to separate out caffeine before we quantify it. So one way we can do this quantification is by first separating the different compounds that make up our beverage, separate those out in a way that we can quantify each one individually. And one way to approach that is using HPLC, or high performance liquid chromatography. So you've already had some experience with gas chromatography. You know that you have usually a mobile and a stationary phase. In gas chromatography, that mobile phase is usually an inert gas such as helium or nitrogen, and a stationary phase which you have mounted on a column. So HPLC is similar to that, but our mobile phase consists of a mix of organic solvent and water. So that can be something called an isocratic mobile phase, which you just have a set concentration of organic and water, or you can have a gradient where you increase or decrease the concentration of organic solvent over time. And then you have a stationary phase. So the stationary phase is a column that's packed with silica, and on that silica we have different chemistries that are attached, and those chemistries will interact with the different compounds in our mixtures to help separate them out. So one of the most common ones you'll see is a C18. So you'll have the silica base, and then you'll have this long nonpolar chain. And then what happens is as your molecules come by this chain, they'll each interact a little bit differently with it. So some will have a higher affinity, and they'll take longer to get off the column, and some will have very little affinity to this nonpolar binding, and they'll be taken off the column really quickly. So by those differences in partitioning in our stationary phase to our mobile phase, we have some sort of separation over time of our different samples. So like I said, we use different phases to separate compounds based on their partitioning coefficient. And partitioning coefficient is essentially saying if I have two different phases, what percentage of a compound would I expect to find in each phase? So if I have something like an oil and water partition, and I put something of mixed polarity in there, what percentage of that compound would I find in the water phase, and what percentage of the compound would I find in the oil phase? And that's just a basic principle saying that different compounds partition differently in mixed polarity systems. So if we put in, let's say, our coffee here, we're going to put in a sample of coffee into our column, and we're going to represent our different compounds that we're to separate based on these colored droplets here. And so let's say we start our mobile phase now, and we have this blue compound. So this blue compound has very little affinity for our stationary phase. It was mostly partitioned into our mobile phase, and so as that mobile phase went through our column, it was taken out first, and so we have this first elution peak that we see on our signal on the right of our blue compound. We'll notice that these other compounds did move along the column, but they're a little bit slower because they have different affinities for that stationary phase. And again, we continue our time here, and now we have our second compound elute. So we had our orange was the second most, or had the second least affinity for our stationary phase, and moved the second fastest through our column and gave us another peak. And then again, now we have our gray compound eluting, and then our green compound. So our green compound has the greatest affinity, so it's probably the most nonpolar compound. If we're going to say this is a C18 column, that means it spent the most time in that stationary phase and took the longest to get off. But effectively what we did is took a complex mixture and separated that out based on its affinity for our mobile and stationary phases to get 4 different peaks here. And these different peaks represent the amount of each compound. And they have different responses depending on the detector, so those peaks won't always be the same size as each other, even if they're the same amount of each compound. So here's an example of a real life separation. So this is actually some of my samples from my hops, and this is what you do to quantify your alpha and beta acids. So we're looking at these main 4 peaks here. You'll see some other peaks too, and they're just some other compounds in the extract that we're not really interested in. But on the right, these 2 peaks are beta acids, and on the left are the alpha acids. And you'll notice these little tails here, these little prenylided tails, and they're really non-polar. So you'll notice that the beta acids have an extra one of these tails compared to these alpha acids, which just have a hydroxyl group here instead. So as we're eluting these compounds over time, we'll see that these ones that are more polar on the C18 column are coming out first. They're coming out at like 9 minutes and 10.5 minutes. And then these more non-polar compounds that have a greater affinity to our column, to our stationary phase, take longer to get washed off the column by our mobile phase. And that's due to this extra prenylided tail here. So this is just kind of an easy way to visualize that different structures and different polarities really affect those retention times of your different compounds. So how are we detecting these compounds? We can show you these peaks, but we need to understand what's generating these peaks. And the way this is actually happening is we have our column here. So we've injected a sample, our column and our different compounds are going to elute at different times. and they're going to flow through our tubing here and make it to a flow cell. And a flow cell is essentially a cuvette with a really tiny volume. So usually they're about 10 microliters in volume, and it's transparent in a way that we can have a source light, so either a deuterium lamp or a tungsten lamp that provides a light source. It's going to shine through our flow cell, and we'll have a detector that will tell us the absorbance of those different wavelengths. And so these are similar to your spectrophotometers that you'll have on your benchtops, but it's kind of recording things over time. So it's recording the change in absorption over time, and that gives us a peak. So if we watch here, we have this compound eluting here. It's been separated, and as that compound goes through, we have this increase in signal over time, and that gives us the resulting peak over time. So now we've run through a sample. Again, this is just that sample chromatogram I had before, and we want to quantify a signal. So what kind of data are we getting out of these peaks? And what we're generally going to get are a peak, or our peak areas. So our software will actually integrate these peaks for us. So we're not going to have to kind of calculate the area of each of these peaks, but it'll take this baseline. So you can see this blue line underneath each of the peaks. And this is kind of saying this is what's zero absorption. So everything above this is a positive signal. And so our software can determine that for us. And it'll identify each of these different peaks and then integrate those to give us a peak area. And so these are just arbitrary units here, but usually it's something like absorbance units times seconds. So you can see each different peak has a slightly different area. In the way we identify these peaks, so if I ran 20 different alpha and beta acid samples, I want to know which peak is which every single time. They're not going to come out as blue, red, green, and yellow. I need some way to identify them from run to run. And the way we generally do that with a UV chromatogram is by retention time. So you already saw this in your GC samples, how the same compounds in the same column will elute pretty much the same exact time every single time you run a sample. So now if we look at these retention times that we've overlaid here, this is how we're going to identify these compounds again in a different sample. So now we would change these peaks from our colors to our retention times in minutes. And now every time we run this, we're going to identify those 4 different alpha and beta acids based on their retention times. And this is important to note because when you're doing caffeine samples, you might have really complicated samples, something like coffee. You'll have a bunch of peaks and you want to know which one is caffeine. And you do that based on retention time. And you'll use a standard to identify what that retention time should be for each compound. So let's take a step back and say, how are we going to actually quantify our samples or the caffeine in our samples, since we have these peak areas now in our samples? And we need to do that by generating a standard curve or a calibration curve. And we do that by using a known concentration of a pure compound. So we take 100% caffeine powder and dissolve it and make dilutions at known concentrations. And then we'll do multiple injections of each of those different concentrations and get an average peak area for each of those different concentrations. So here's some just random data I generated to simulate what a standard curve might look like for a chromatographic run. So if we have our different concentrations of standard, units don't matter here, but we have 50, 100, 150, 200, and our different average signals in peak area. So if we plot our standard units versus our peak area, then we can get this standard curve by fitting a linear trend line here. All right, so now we have this linear trend line for compound X, and we want to quantify the compound X in our complex sample. So we run our chromatogram. We have our peak area for our unknown sample, and that peak area is 1153.2. So what do we do with that peak area? Well, we're going to use this calibration curve to calculate the concentration of that compound based on our standards. So we'll use that equation from our fit line, plug in our peak area as y, and solve for x. And we'll see that x is 140.9 units. So now we have a concentration of this compound x in our unknown, where before we just had a peak area. And if you want to visualize how we did that, we're going to go over from our absorbance units, which is our y-axis, to our standard curve line, and where we hit that, we'll drop down to see the concentration of our standard. And that's how we solve for an unknown concentration. So in today's lab, like I mentioned, our footage is limited, but we will look at a few screenshots, go through some of the footage, and then cover how to interpret a report. So the first step, we're going to look at how to set up an injection in the HPLC software. So before we inject our sample, we need to tell the HPLC how we want the data stored and labeled, and what sort of output we want. Then we're going to have a video covering how to actually do an injection. So we'll be doing manual injections with a syringe. Then we'll watch peak elute over time, so we'll actually see the separation. Then we'll go over how to interpret a run report. So every time you run a sample, you'll have some sort of data generated in some sort of report. So it'll be a graph of our peaks, and then giving us a table of retention time and area, and we'll cover how we're going to use that data to find the concentrations in our standards and in our unknowns. Then for your lab, you're going to be generating a calibration curve using raw HPLC data that we'll provide you. And then you're going to be determining the caffeine content of different beverages using raw HPLC data. And we'll be doing name brand cola, a store brand cola, instant coffee, and hot tea. And we'll give you all the raw values for those different runs, and you'll calculate the caffeine content in each of those beverages. Things to consider while you're watching this lab and listening to this lecture. Think about what's the purpose of this activity. What's our research question here? Why do we need to perform HPLC to separate caffeine in a sample? And what's your hypothesis? So if we have our 4 different beverages, based on your knowledge of those beverages or based on their labels, which ones do you expect to have more or less caffeine? And how closely do you think the values we determine in lab are going to match the label content of caffeine? And like I said, why do we need to use HPLC to quantify caffeine in our beverages? Why can't we just throw it in our spec? And then, of course, what sources of error would you expect to find in this type of analysis based on the protocol provided? So this is actually a quantitative method. We're taking a raw sample, we're separating out our compounds and quantifying that unknown based on a calibration curve. So there are a lot of different preparatory steps that can lead to error in our final result. And that comes from sample prep and goes through to our data analysis and how we present that data. So think about that both through this pre-lab discussion as well as one of your lab questions. So this is what the software interface looks like for our HPLC. This is Open Lab software from Agilent. And some of the first things we want to look at, there's a lot going on in the software, but on the left here we can see all of our different method files, and the method file we're using today is caffeine. I made it, so it's BJC, and it's about 2 minutes long. So we need a descriptive name to differentiate our method from all the other methods saved on the software. If you work in a lab doing a bunch of different analyses, you'll have a bunch of different methods set up. So you need to be descriptive in your labeling and make sure that you have an explanation of what each different method is in your lab notebook when you do this. The next thing we want to look at is this grad pump window. So this is a gradient pump. And so we have 2 different pumps, pumps A and B, and they can each pump individually, supplying different concentrations of A, which is our water, and B, which is our methanol. So right now this is set to be 65% water and 35% methanol. And this is an isocratic method, so this means it stays at 35% methanol throughout our whole run. Over here, we have our flow rate. So our flow rate is 1 mil per minute, and that again stays the same the whole run. And we can monitor pressure. So this indicates A, whether or not our pumps are pumping and we have flow through our column, and B, it can be an indicator of any leaks. So if we have a normal operating pressure and then suddenly we see a drop, that could indicate that we have a leak somewhere in our system. Or C, if we have a spike in pressure, that could mean that we have something stuck in our column or that we have contaminants building up in our column over time. So we need to stop and diagnose what's going on there. Our next window is our diode array detector window. So right now we can see this is set to 278 nanometers. That's the absorption maximum for caffeine. And our light is currently warming up, so it's not currently ready. Before we start this run, we want to make sure that this is green and our lamp is warmed up. We also have this button here, and this is just to adjust the baseline. So after our lamp is warmed up, we press this button, and it'll just readjust this signal to a baseline of 0 milliabsorbance units. That really doesn't affect our integration, but it's just kind of simpler to see peak height if you're starting at 0, and then your peak goes up to however many milliabsorbance units. There are a number of different options in the software that we don't really use for this lab, but you do have things like run queue or easy sequence, where you can set up a number of different runs and label them what their identity would be, and set up an order in which they'll run so that you can more simply just inject each sample and have the data stored in the appropriate location. You can also have an HPLC with an auto sampler. So if you have an auto sampler, you need to tell that auto sampler the sequence of which vials it needs to draw a sample from, how much it needs to draw, and in what order. So after we have our HPLC set up, our pumps are green, our lamps are green, the instrument is ready, we're going to go to run control. This is where we'll input our sample info. So it'll open up this window, our sample info window, and this is where we need to tell the software what our sample is called and where it's going to save. And we need to change this for every sample we're injecting, because every different sample has a different identity. It belongs to a different lab group and a different day. So we can identify where it's going first. So we'll start in the software and in the data file. Our subdirectory, we made a folder for this course, so that's Food Science 410. And then some information that we're going to put in here. We're going to have our date and then our sample name. So every time we run a sample, we'll know which day it's run on, and then we'll know the sample name. And we'll give it a descriptive sample name as well. So we have 410 for the class. W is Wednesday, the day of lab. 2, this is probably our group number. The identity of the sample, this is a calibration sample. Our concentration that we're running, as you know, we're going to have 5 different concentrations. We need to label which one we're running. And then last, we have our rep. So if we're doing 3 injections, we need to know which replicate we're on. You can also put information here of how large their injection is, and in this case, we're doing 10 microliters. So once we've adjusted each of those parameters for each run, we can come down here and click Run Method. So this first sequence here is preparing an injection. So as you can see, these marks on the side of the needle are in microliters. So I've drawn up the caffeine sample a little bit past 10 microliters in order to make sure there's no air bubbles in the syringe. And now I'm going to slowly press out the excess sample until I get down to as close as I can to 10 microliters. So you got to think, like, if I'm off by just 1 microliter, then that's 10% error from injection to injection. So I need to be really careful to get that correct. Now I want to move this lever to the load position. So we're loading our sample now. And I'm going to go to that second stop, quickly inject the needle, and then turn this lever to inject. And then what that does is moves our mobile phase from running past the sample loop to through the sample loop and loading our sample onto the column. So now our run is started, and we're going to watch our sample elute here. So here you can see this first little peak. I apologize for the quality. I'm holding the phone here and trying to use a stabilizing effect. So this is that little impurity. We'll see later. And then just about now, we're going to start seeing our caffeine peak eluting. And so you can imagine some packed caffeine is just coming off the column now, and now we're getting to the main bulk of that caffeine elution. So we have a really large peak here. And here, let me rescale it. And we can see this peak form. And now the peak's going back down, so just the last of the caffeine is getting off our column. And we have a really nice clean peak for caffeine here. And we would repeat this for all of our different calibration standards and concentrations, as well as our samples. So this is a really short method, it's just about 2 minutes. And then once this is done running, we'll see here in a minute, the software is going to generate a report for us. And this does this every single time you finish an injection on manual mode. So it gives us our data right away. and we can see what peaks we're eluting and how large those peaks were and what the retention times were. So here's this automatically generated report. Now we'll get into analyzing that. So as you saw, we eluted our caffeine peak, and now our software generated a report for that run. And this is where we're getting our raw data from. So every single time we run a sample, we're going to generate one of these reports from the software. And here we can see the important information we need. So we have our data file directory, and then what we want to look at is our sample name. So make sure that this is the sample that we just input into our software and not a different sample. So yep, this is our calibration standard, 250 parts per million, our second replicate. And this is our raw chromatogram. So here we have our milliabsorbance units, and we have our peaks. And these numbers here are our retention times. So you can actually see we have two different peaks here. Even though this is a standard, there can be impurities, whether that's in the caffeine powder if it's not 100% pure, which very few standards are, or if there is some sort of contaminant in the needle. Sometimes you get peaks that are not your standard when you run a standard. But based on the quantity of caffeine that we're injecting, 250 parts per million, that's pretty high. We're going to go with this as our caffeine peak. And since this is a calibration standard, before we run our future samples, we want to note this retention time. So here it's 1.229 minutes. So every time we run a sample, a peak at around 1.229 minutes should be our caffeine peak. So this is where we determine our retention time, is in our standards. All right, so now we've copied down that important information. We're going to go to our table. And this is further down on the report. If you want to find it on Canvas, you can follow along. And here we have our data table. So like I said, this is a pretty simple chromatogram since it's a standard. We just have two peaks. And we have this peak table. So we have our peak number, and these are just in chronological order. And we have our retention time, which we use to identify our peak. And then the other things we care about pretty much are just the area. And sometimes you care about percent area if you're doing a complex mixture. But here today, we're just going to worry about area and retention time. So we're going to go down to our retention time for caffeine, which is 1.229 minutes. And our peak area. So we have 8,813.81250 milliabsorbent unit seconds. And this data we would copy down to our Excel file. And we want to take that data, so we want to put in the concentration of our standards, 250 parts per million, our retention time, 1.229 minutes, and then our area in a different column. And recording all that data, we'll put that all in the same Excel file and then use that raw data to generate our standard curve and to identify caffeine concentrations in our unknowns.